this class moving a little quickly for you?
No.
No? A little bit? One of the reasons why is that there's a holiday on Monday, campus is closed, and then we have lecture exam on Wednesday. We don't have any content being delivered next week, so there's no week 5 PowerPoint. Holiday Monday, lecture exam 1 on Wednesday. Everybody got it? Okay, one of the reasons why. I lose enough days. This is basically what I'm going to cover this week, gets us through 5 weeks. Already had one lost day and holiday already. This is the second holiday and lost weekend, so that's why we have to do this class. All right. Again, next week schedules, all we're gonna have next week is lecture exam 1, and that's it, no labs next week. But we do have lab 3 this week. Okay, we have gone through metabolism, and we started on DNA. There were 2 subtopics of DNA. One is protein synthesis, and the other is DNA replication and cell division. We're almost done with the protein synthesis material, and so we'll finish that up, and then we'll go on to, we'll actually start lecture exam 2 material today as well. All right, so a couple of subtopics to, well actually we already covered one, and that was gene regulation. And remember with gene regulation, I had several slides on the lac-operon theory. You only need to know that the lac-operon theory of gene regulation is an example of repression or negative control. You do need to know the slide that deals with cyclic AMP because we will see cyclic AMP later. Okay, the rest of the slides don't worry about, but you should know that about the lac-operon, repression, negative control, and the way a cell can decide whether or not to expend ATP, depending whether there's glucose or lactose involved. And that's by using adjusting levels of cyclic AMP. All right, the other subtopic on protein synthesis is this, mutation. Go ahead and fill in the blanks.
I'm already lost.
Everybody got blanks? In biology, when we talk about mutations, we are talking about changes, random unintentional changes in DNA nucleotides, okay? Now again, we have about 3 billion base pairs, and because of the purines matching up to pyrimidines and having to have the right number of hydrogen bonds, does this sound familiar? The base pairing is remarkably accurate. And out of 3 billion base pairs, every time your cells replicate the DNA, maybe only 1 will actually be misread when it makes copies. 1 out of 3 billion. That is an incredible level of accuracy. Everybody got that? Now, most of the mutations are what we call neutral, which means nobody knows that it even happened. I'll explain why in a bit. But before we get that, there's a couple of highlighted words here. We're going to spend most of our time talking about somatic gene mutations. So I need to go over these definitions. There's one example of a germline. First, you got to know these terms. Germline mutations involve all your cells, including your gonads. And if a mutation involves the cells of your gonads, that means the mutation is going to be carried over into your spermies and Aedes. which means you can pass that mutation on to your kids. That's a germline mutation. Everybody got that? Somatic mutation means it doesn't involve the gonads, and so that's just your problem. You do not pass somatic mutations onto your kids. Good example of that would be cancer. Cancer is actually a multiple mutations, but that's yours. You don't pass on, well, there's a couple of really rare exceptions, but by and large, you do not pass on cancers to your kids. Everybody got it? What you do pass on to your kids is your immune system genes. And if you have an immune defect for fighting cancer, there's a chance you have passed on that immune defect to your kids. That's why you go to the doctor, the medical history report always asks about family history about cancer. They want to know if your parents or your siblings have had cancer, because that means there's a chance that there's an immune defect in your genes. Everybody got it? But it's not like you pass the cancer directly to your kids. Everybody got it? So this, what you want to know is this gets passed to your kids because it involves the gonads. This does not, because it does not involve the gonads. Second, there are gene or points, and sometimes they call these gene mutations. I use gene up there. Sometimes they call them point mutations. And that's versus chromosomal. Basically, point or gene mutations, and we're going to spend most of our time talking about this. Point or gene mutations is 1 to 3 nucleotides. Got changed. And you cannot see, if you take a picture of the chromosomes, the karyotype with the chromosomes and metaphase, you can't see them. You can't see 1 to 3 nucleotides. It's just too tiny. When you're talking about chromosomal mutations, you're talking about changes in the hundreds of thousands to millions. You could see this. Can anybody give me an example of a chromosomal mutation that everybody knows? Down syndrome. Trisomy 21. There's 321 chromosomes, and they're numbered. So you get down to, and they're all paired. You got 2 number 1s, 2 number 2s, 2 number 3s, 2 number 4s. You go to number 21, and there's 3 of them. You can actually see it in the karyotype. That's because the chromosome 21 has millions of nucleotides. So it's a matter of scale. You do a chromosomal mutation, you can see it. It's huge. Gene or point mutations you cannot see. Okay? And again, when we talk about these types of mutations, somatic gene mutations, we usually don't even know that they happen. And the reason why was already given last week when I talked about how less than 2% of your DNA actually codes for anything. If you get a change, a point mutation of 1 to 3 nucleotides in space filler, your cells don't know the difference. It's neutral. Everybody get that? Now this is where I'm going to expand on it. Are you guys tracking with me? Track with me. If you're not tracking with me, you have to make sure you are. And that's on you. Okay? You're grown-up students now, you're not kids. If you're not tracking, it's on you to make sure you get back on the ballpark. Okay? Because believe you me, you get in that program, pharmacology is going to be your separator. You flunk it, you're out of the program. I'm trying to get you up to this level so that you don't flunk it. And I'm not going to do that by slowing down and oversimplifying things even beyond what I'm doing now. Okay? All right. Even if one of these point mutations, say it's 1 nucleotide, happens even on the gene, it may be neutral. And this is because of codon redundancy. This is the 3rd bullet. If you look at the table of codons, which you do not need to memorize for lecture exam 1, what's important that you do is you will know that codons are on messenger RNA. It was on the whiteboard last week. Code is DNA. Codon is messenger RNA. Anticodon is transfer RNA. You've got to know that. You don't have to memorize what they code for. Remember, it is 3-to-1 ratio. Do you guys remember that from last week? 3 nucleotides per amino acid. Well, here is the table of codons. First nucleotide, second nucleotide, third nucleotide. Maybe write that down somewhere. This is the first, this is the second on top, this is the third on the right side. And each of that combination of 3 RNA nucleotides, messenger RNA nucleotides, because this is codons, is for 1 amino acid. So far, so good?
2.
Codon redundancy is this. If you look, say, at the amino acid proline, right here in green, everybody see that? Notice that it only codes by the first 2 nucleotides. The third one is irrelevant, save that the third one has to be there. As long as it's there, the third one, it doesn't matter which nucleotide it is. Look at that. If it's C and C, you're going to get proline. It doesn't matter what the third one is. It's going to be proline. You can have a point mutation on the third nucleotide. It's the same amino acid, which means the protein is unchanged. And your cell doesn't know there was a point mutation there, even when it happens on a gene. Are you following this concept?
If the mutation is on the first or the second, though,
Then it changes.
then it changes it?
Then you're gonna know something's messed up. But then it's not even for all of them. If you look at it, only a few amino acids have all 4, where the third one is, just have a third one. It doesn't matter what it is. But some of them, is only split into 2. Here, like here. So if it goes to U or C, okay, it won't change. But if it goes to A, it's gonna change. Can you see that in the table?
So is it basically just the nucleotide bases that affect it? The amino acid doesn't have anything to do with it at all?
No, it's the opposite.
Oh.
For the third nucleotide, it doesn't matter which nucleotide it is, it'll be the same amino acid. And that's why you've got to get this thing straight first. Code, codon, and a codon primary protein structure. That's how you're going to make sure you're going in the right direction on this. Yes?
Is that what makes AUG so special?
AEG is special in that it codes for methionine, right here, and you can circle this right here. This is the initiator codon. Methionine is always the first amino acid in the initial protein. So if you do get a point mutation in the third nucleotide, it may not change it. And if you look at the numbers here, it's 8 out of the 20. 8 out of the 20, you can change the third one, it won't matter. But the other 12, you change the third one, you're still going to change the protein because you're changing the amino acid. Everybody got it? I need to go kind of move faster here, or we're going to fall behind. All right, so if you do get a different amino acid, even if it's just one, it could have devastating effects on the overall protein because of specificity. If you want a good example, let's talk about sickle cell anemia. And this is the one example. This is sickle cell anemia is actually a germline point mutation. People do pass these on. To the kids. But it's only one nucleotide that changed, and notice here, it's the second one that changes. So because the second one that changes, guaranteed that's going to change the amino acid, and it changes from glutamate to valine. Okay? Then look at the numbers. And I think I talked about this last week when I talked about the 3-to-1 ratio, how in hemoglobin it's like 300, so it's 900 nucleotides. Do you guys remember me saying that? It's not 300, it's 287. But I rounded up. okay? You're only changing one amino acid out of 287. The problem with this is the valine sticks out on the outside of the protein. And when you change it, and the oxygen levels drop, one adjacent hemoglobin, I think it's the beta chain, there's two alpha chains, two beta chains, will also have valine sticking out, and valines stick together when oxygen goes low. So there's another one, sticks together here, there's another one, sticks together here, and they start sticking together. Because the valines stick on the outside when the oxygen levels get low. But it won't form a straight line. This line has a slight curve in it. And when all the hemoglobin in the red blood cell does a slight curve, you get these sickle cells. Now, does the name make sense?
Mm-hmm.
From one nucleotide out of nearly 900. But because it's in the gene, and because it's in the second of the codon, it's a different amino acid and it has devastating effects. Everybody get me point mutations now? If it doesn't happen in a gene, you never know. If it does happen in the gene and it's the third nucleotide, you may still never know if it's one of the 8 out of the 20.
Does sickle cell only happen in the second one, or can it also happen in the first?
And in this case, it's the second one.
Okay.
Yeah, you're not gonna get sickle cell anemia if you don't change it to valine, because that's the thing that causes it, is the valines stick together
Okay.
from different globs. Okay? And this is what it looks like. When the whole thing forms these great, long, slightly curved polymers, the cell looks like that, and they get stuck in capillaries. There's no way that thing's getting through a capillary. Okay? Any questions? All right, that is protein synthesis. So I went over the meat and potatoes of it last week, covered that one thing on gene regulation, covered this thing on point mutations. I'm telling you people, there is way more than this. I'm giving you just a tiny bit. All right, the last topic of lecture exam 1 is on DNA replication and cell division. Remember that we only have 46 double helices. That includes the proteins in a dividing cell. You're gonna need certain enzymes to get this going, and I think I need for you to now turn to page 16 in the lecture packet. We're going to use pages 16 and 18 today. We already did 17, that was the last topic. All right, here it says certain enzymes have to unzip the double helix. If you look on the bottom of page 16, that enzyme is helicase. Is everybody on the bottom of page 16 on the lecture packet?
Mm-hmm.
The enzyme that unzips the double helix by breaking these hydrobonds is helicase. And you might want to write that next to the second bullet, since it's in writing there on page 16. So the double helix becomes 2 single strands. Now you're going to add complementary DNA bases to those intact strands. It looks very similar to transcription. In fact, if you look at the animation from last week's PowerPoint, this animation looks exactly the same.
Mm-hmm.
But it's not. Because in that one last week, all the bases were RNA nucleotide bases. These are all DNA nucleotide bases. But it is still hydrogen bonds first with the right size nucleotide. It's got to either be the purine or pyrimidine, and it's got to have the right number of hydrogen bonds. Hydrogen bonds form first, then the covalent phosphodiester bond forms second. And notice also, These are all triphosphates. Can you see that? Which means to duplicate 3 billion base pairs is going to take your cell 6 billion ATPs.
Damn.
Do you see why having mitochondria, making a ton of ATPs is crucial? 6 billion ATPs to replicate your chromosomes.
That's some busy work.
Okay? Now the enzyme that is driving this is DNA polymerase, and the specificity here is that the sugar's got to be deoxyribose. If you look on page 16, it's RNA polymerase on top. It's still helicase that unzips it, but it's RNA polymerase, and that's going to be specific to get nucleotides with ribose as a sugar. Here it's going to be deoxyribose. Okay. Now, this was covered in anatomy, wasn't it?
Yes,
Semi-conservative replication?
yes.
Okay. But here's going to be the difference that you need to know for physiology. DNA polymerase, the enzyme, can only work on the original strand from the 3-carbon end to the 5-carbon end. It can only go in that one direction. In fact, write that next to the slide. DNA polymerase can only work on the intact strand, going from 3 apostrophe to 5 apostrophe. DNA polymerase can only replicate a new strand against the original strand on that direction on the original strand. 3 apostrophe to 5 apostrophe. And because the two strands are not mirror images, they're all left-handed, this side is going to do this in one intact, what's called a leading strand. What's this guy got to do is it's got to go boop, hop over, boop, hop over, boop.
Yeah.
Because it can only go from the 3 apostrophe toward the 5 apostrophe. Boom, hop over, boom, hop over, boom.
When you're saying hop over, is it splitting, or what do you mean by hopping over?
It's going to go until it runs to the last strand that was made, jump way over, and come back in that same direction until it gets to the last strand that was made. Boom, jump way over, then go toward the last strand that was made. Boom, go over and go toward the last strand that was made, but it has to go in this direction.
The enzyme?
Yes,
Okay.
the DNA polymerase is the enzyme. It has to go from 3 apostrophe to 5 apostrophe. This is what is referred to as the lagging strand, and the reason why it was found out is some Japanese biologists found these in a dividing or a cell that's doing DNA replication, these DNA fragments. And they called those after the guy that discovered it, Okazaki fragments. That is what is called the lagging strand. When we first learned the semicircular, semi-conservative replication, it was like this in anatomy, where both strands were leading. That was a lie, an oversimplistic lie.
Okay.
The truth is, DNA polymerase can only go in one direction, and because the two strands aren't mirror images, one looks completely different from the other. This animation is a lie.
Okay.
In fact, write that next to the bottom left of this slide. This animation is an oversimplified lie
Lies.
that we learned in anatomy. Ain't good enough in this class.
Yeah, Hara is a liar. Okay.
Now, even though you're going to get Okazaki fragments on the lagging strand, and this is all described on the bottom of page 16, there is a third enzyme that links all those fragments, Okazaki fragments, together, and that's called DNA ligase. That makes it all one intact strand. That's on step number 4, the bottom of page 16. And the net result is this: 2 complete DNA double helices, which at the most might have 1 out of 3 billion base pairs misread. And that's at the most. There's a lot of times where it's 3 billion out of 3 billion are right. Okay? And this bottom line is what's called semi-conservative replication, and that's because every time a double helix replicates, both of the original double helix strands stays intact. That's in gray. Look at it on top. It's in gray. They don't go away, they're intact. And the cell is adding a new purple strand to it. So each new daughter double helix is half old, half new. Semi-conservative replication, yes?
Is there always gonna be a leading strand and a lagging strand?
Yes. Every time your cells replicate their DNA, getting ready to divide, go through cell division. But by the time it's gone through the DNA ligase, you can't tell that it had to have a lagging strand. They look identical. Everybody got it?
So helicase is what unwinds it.
Unlines it.
DNA polymerase is what goes to the other side of the strand and adds.
Is what is adding a complementary strand from a 3' to a 5'.
So is helicase only on the 5 to 3 strand?
What's that?
Does helicase only unwind on the 5 to 3 strand, on the leading?
Helicase is the
Or does it just cut?
is the unwinding enzyme that's breaking the hydrogen bonds
Okay.
between the strands. It's the same helicase for you guys that is also unzipping the strand in the gene for protein synthesis. And that's up on the top of page 16, number 1 on the top. So everybody understand page 16? It's comparing protein synthesis that we covered last Wednesday to DNA replication that I'm covering right now. Everybody see that? And in both cases, the unzipping is helicase for you guys. Again, that is a lie.
Man.
I mean, how much of this do you guys want me to do? This topic of DNA replication will be probably 3 lectures, if you want the whole story. You want the whole story?
No.
Okay. So just accept the fact that I'm lying to you.
Good enough.
Like I'm some sort of player or something. Just lie to everybody. Any questions on semi-conservative replication? All right. Now, when does the semi-conservative replication happen? Well, we have to look at the overall concept of cell division. And here again is the concept of germline versus somatic cells up there on top. You must know that germline cells do both mitosis and meiosis. Somatic cells only do mitosis. That does not mean that germline cells don't do mitosis. It means germline cells do both. But somatic cells only do mitosis. Oh, females, by the way, their mitosis ends, I think, before 6 weeks of embryonic development. They're already done with mitosis. Their germline cells don't do mitosis anymore. Man, we do mitosis until we're pushing up daisies. Okay, when we talk about cell division, I do need to talk about these 2 words. In this context, kinesis, it means division of. Karyo is nucleus. Cyto is cytoplasm. Second bullet is just mentioning, out of all the complexities of this, that karyokinesis happens first, followed by cytokinesis.
Okay.
If you remember IP math, the cell cycle, I'm going to cover here in a second. I'll go back over this if you need to. But karyokinesis actually starts happening in anaphase, finishes in telophase, and cytokinesis comes after telophase, or at the very end of telophase. So that's why karyokinesis first, cytokinesis second.
Okay.
And the cell cycle, I teach my anatomy students, is IPMAT. Which, if this was more accurately, chronologically accurate, I would call it PMAT. Cells in interphase, the vast majority of the cycle. PMAT is a tiny slice of the pie. If you look at this picture, interphase is purple. And they expanded the size of mitosis to fit the words in. The actual slice of the pie for mitosis is much skinnier than that. Everybody got it? And it's just cycling around I, PMAT, and here you can see that PMAT is mitosis. The DNA semi-conservative replication happens in the middle of interphase. Okay? That's tying what we were just learning about. Your textbook gives cytokinesis its own stage in this, but then IPMAT doesn't work, so that's why I don't do it. I just put cytokinesis at the end of telophase, which a lot of textbooks do. Okay? All right, let's look at interphase, since that is the vast majority of the pie. Starts at G1. This is where the cell needs to grow physically, because the original cell just got divided into two, so the smaller cells. So you're gonna increase how much you're bringing into the cell in pinocytosis. And then you gotta fill that fluid up with important things like proteins and RNA. And the centrioles will actually replicate right after cell division. Remember, the centrioles set up the poles for the dividing cell? Well, when the cell divides, each one only has one. Okay, one pair. But you need two pairs to divide the cell, so that'll replicate right away. S is when that semi-conservative thing happens. And then G2 is when the cell's getting ready to divide again. Other metabolic activities slow down as it gets ready to do that. Now there is a thing called G0 for cells that either lose a nuclei or fuse, so it has multiple nuclei. Those cells do not divide anymore. Even cells that just have one nuclei, like neurons, a lot of those don't divide anymore. We used to think that none of them divided. That's not true. There are neural stem cells that divide. But once it's functioning and synapsing and firing action potentials, they don't divide anymore. Okay? You can only get rid of them on a Friday night drinking bench. Okay. We gotta talk about a lot of Chrome words here. Okay. We're going to cover mitosis first. And when the cell is going into prophase, the P of PMAT, the chromosomes are going to start to condense. When it's diffused, in the interphase, we call it chromatin, because we can't see 46 chromosomes. It's all intermixed, it's diffuse, so we call it chromatin. Spelled just like this except for an N. Then when it starts to condense in prophase, it'll look like an X, like what I drew up on the whiteboard, what's on the screen here. And the reason why it looks like an X is because the chromosome has already gone through semi-conservative replication. There are 2 copies of each chromosome. Everybody got it?
Mm-hmm.
Each copy is called a sister chromatid. That's the first arrow. They are connected, each copy is connected at the centromere. What I would write next to centromere is that if you have one centromere, you have 1 chromosome. If you have 1 centromere, you have 1 chromosome. Here I've drawn it as a black circle. How many chromosomes have I drawn on the whiteboard? 1. But there are 2 copies of it, and each copy is called a sister chromatid. Everybody got it? It's still 1 chromosome. When the cell gets ready to go into mitosis, the centriole pairs, and there's 2 pairs, establish the poles of the dividing cell. And radiating out from those centriole pairs are these microtubules called the spindle apparatus, and I've drawn that in green. When it gets ready to do mitosis, one pole hooks up on one side of the centromere, the other pole hooks up on the other side of the centromere. Okay? Everybody got these terms? This was covered in anatomy, wasn't it?
Mm-hmm.
I'll go ahead and just fill in the blanks for these.
I remember crying over all those C words. I like that animation.
Again, the chromosomes start looking like little X's in prophase because they condense into visible chromosomes. The nuclear membrane or envelope disintegrates, and the centriole pairs, there's 2 of them, they migrate to opposite ends of the cell, establishing the poles. They tie the spindle apparatus from the centriole pair to each centromere, one on one side, one on the other, and then they shorten. As they are shortening, the chromosomes are continuing to condense. As they shorten, they pull all 46 chromosomes at the equator. Meta means middle. When all the chromosomes are in the middle at the equator, we call it metaphase, and the chromosomes are maximally condensed, which is why we take pictures of them in metaphase. At metaphase, all the chromosomes will have a consistent appearance because they are all maximally condensed. Does this make sense?
Mm-hmm.
They keep pulling. The spindle apparatus keeps pulling until it actually splits the centromeres. Now, what did you write on the previous slide? 1 centromere, 1 chromosome. How many did I draw right here? How many are drawn here? Two. At anaphase, the number of chromosomes doubles, and it has to double. Because if you're going to divide in this plane at cytokinesis, you want each daughter cell, and you're going to have two of them, to have the same number as the original cell. If there's one here, you want each of these two new daughter cells to have one each. If you have 46 here, you want each of the new daughter cells to have 46 on each side of that dotted line. Does that make sense?
Yes.
And then telophase is basically the opposite of prophase. The chromosomes decondense and form nuclear envelopes around them. By the way, this first bullet is karyokinesis. Look under the telophase box. The first bullet is karyokinesis. That happens first. Then the second bullet, cytokinesis, division of the cytoplasm. Right on this plane.
Hold on, you're saying the first bullet,
It's
which is,
karyokinesis.
and what bullet are you talking about?
There's two bullets in telophase. The first bullet is karyokinesis that happens first. The second bullet is cytokinesis, which is what I told you.
Okay,
Karyokinesis happens first, follows by cytokinesis.
okay.
Okay? But there is a brief moment in time, and again, this is a tiny slice of the pie. And now this is just a tinier slice within that tiny slice. That cell has 92 chromosomes. For a brief period of time until cytokinesis, and boom, each of the two cells have 46.
Okay.
Okay? And that makes sense. If you're gonna go from one cell to two, at some point, you have gotta have 92 centromeres, 92 chromosomes, at some point, right before cytokinesis. Any questions?
Okay.
Okay. I like this figure from the old 4th edition. I don't know why they got rid of it. And you can see there, I'm giving you oversimplified lies. I'm not even giving you pro-metaphase. Okay? It's oversimplified lies. And then this computer doesn't play my animations anymore. So hopefully you can get access if you run the PowerPoint on the Pearson website to get. They will play the animations because they're pretty good, and I really like playing them because I have to stop talking. Even for 40 seconds. All right, are we ready to talk about meiosis? Any questions on mitosis? Okay, meiosis. This is how we make spermies and eggies. And instead of IPMAT, it's PMAT IPMAT. What does that tell you if it's PMAT IPMAT?
Happens a second time.
There are 2 rounds of divisions, okay? And we gotta get our spermies and eggies to be starting out diploid or 2-end so that they only have 23, N or haploid. In biology, the N is the number of types of chromosomes in our species. Homo sapiens have only 23 types of chromosomes. That's our N. Our regular somatic cells have a set from mommy and a set from daddy. So our regular body cells have 23 pairs, what is called 2N or diploid. There are other species that routinely in their life cycles go tetraploid, okay? Biology is extremely complex. You're only learning a small slice of the biological world when we're doing human anatomy and physiology, okay? We only go between diploid and haploid is our spermies and eggies. So that way, when you bump uglies, your child will have the same number of chromosomes as you. In fertilization, it goes back to diploid. Any questions? Okay. Again, meiosis has to do with the germline cells and making haploid sperm and oocytes. And it involves 2 cell divisions, producing 4 haploid nuclei. Actually, on this slide, next to the slide, write male meiosis. This is not meiosis overall. I'm teaching you male meiosis here, lecture Gen 1. Because males are better. Because males are simpler.
Yeah.
The end of the semester, we'll do female meiosis, which is incredibly complex, relatively speaking. All right, so let's look at the karyotype. Remember, we take a picture of the chromosomes at metaphase because they are maximally condensed. When we do that and then arrange them from biggest pair to smallest pair, that's a karyotype. You can see the pairs here. And they're numbered by size and what are called autosomes. And again, if this person had Down syndrome, there would be 3 of these. That would be a chromosomal mutation. You can actually see it in a karyotype. All right, the pairs are called homologous chromosomes because they come in pairs, so the 2 number ones are homologous. They're sometimes referred to as homologs for short. The first 22 pairs are called autosomes. The 23rd pair are called the sex chromosomes, with females being truly homologous XX and males having this X and Y, this Y being this stunted, retarded little piece of DNA,
Oh my goodness.
which has no genes crucial to life. How do I know there are no genes crucial to life on the Y chromosome? Critical thinking people. Yeah, there would be no chicks. If there were any genes crucial to life on the Y chromosome, there'd be no females, because females don't have one.
I don't get it.
What sex is this person's, that the karyotype is taken from?
Male.
Male. Down here, 23rd pair, XY. Everybody see that? Okay. Here's the thing about the XY. Even though they are really dissimilar, in meiosis, they act just like homologous pairs, like the 2 number ones, which are identical. They still act as homologs, even though they're dissimilar. Everybody got it? All right, let's do meiosis 1 first. This is the freaky one. And we can call this the reduction division because it is going from diploid to haploid in the first division. Reduction division. Go ahead and fill in the blanks here. Okay, in prophase one, some of it looks similar. The chromosomes condense, nuclear envelope dissolves. But the difference here in this freak we call meiosis one is the homologs actually get really close together. This is the only division where that happens, okay? Well, for one thing, in meiosis two, there are no homologs. They're all single chromosomes, but in mitosis, the two number ones are just random. Wherever they are, it's just completely random in mitosis. Not in meiosis one. The homologs get really close together and actually exchange pieces. The exchange of pieces is called crossing over, that I've drawn here. Okay? They are maximally condensed. And align and dequare and metaphase one. Oh, and the other thing with prophase one is the spindle apparatus attaches differently. Instead of having two attachments on a single centromere, in meiosis one homolog will go to one pole, the other homolog will be attached at the other pole. Each centromere only has one spindle apparatus connection, not two. Does everybody see that? Does everybody see that? So that in anaphase one, where the shortening spindle apparatus here split the centromeres, because each had two connections. Here, since they only had one, they do not split. Everybody see that? In other words, look at me. X's stay X's. In mitosis, the X's became lines. In meiosis one, X's stay X's. How many chromosomes do you see here?
2.
How many chromosomes do you see here?
2.
There was never any doubling in anaphase one, and you don't want it to double because you want to cut it in half. This is the reduction division. You want to go from 46 to 23. And the way you do that is you don't split centromeres. Okay? Centromeres do not split. The entire X moves toward either pole. Everybody see what that means?
Yes.
In fact, you would say that. I would write this. Homolog looks like an X. Telophase one, you do not have a reversal of prophase. Why bother? You're just going to go into another cell division. So they don't uncoil and no nuclear envelopes form. But one cell goes to two, you do have cytokinesis. You do have karyokinesis, you just can't see it. The PMAT, IPMAT is because after telophase one, you go into interkinesis with the two haploid cells getting ready to go into meiosis two. Everybody got that? Cell division is a really good matching section on lecture exams because it comes in 5. Some in 4. All right. Meiosis 2 is very similar to mitosis, but it's doing so with half the chromosomes. X's become line, but you only have 23 of them instead of 46. Okay? Again, this is male meiosis. Any questions? So on this animation goes really fast. When you see one big black circle going to 2, that's meiosis 1. Where you see 2 going to 4, that's meiosis 2. Notice that when 1 goes to 2, X's stay X's. But when 2 go to 4, the X's become lines. Meiosis one, X's stay X's. Here's a crossing over. Boom, they stay X's. When it goes from 2 to 4, X's become lines.
Crazy.
Just as I explained. The original cell has 4 chromosomes, 4 centromeres. When it does that, it's only 2 each. When it does that, they're also 2 each. Everybody see that? The reduction division is here. 4 in the original cell. Only 2 in each of these 2 cells. That's a reduction division. Then it just goes to 2 to 2. Any questions on mitosis and meiosis? This is from your textbook showing you again, this is male meiosis. Female meiosis doesn't look anything like this. Okay? Would you like me to talk about lecture exam 1 now?
Yes.
And then we gotta get going on lecture exam 2. At least get started on it. All right, here's how it's gonna happen on Wednesday. You guys ready? You're gonna come in with your small blue book, and I mean small. If you come in with one that's 8.5 by 11, consider yourself an open target for me giving you some shit. Well, I'll shame you for bringing a big one. Bring the small one. You're gonna drop off a virgin blue book on the front desk. Don't write anything in it, including your name. It is completely blank. You drop it off in the front desk, go find a seat in the room. I think we should be able to have an empty seat against everybody. So nobody sits right next to each other. If I do run out of seats, the only row that you can sit right next to somebody is the front row. You're gonna put your cell phones and watches, because there are smart watches. So even if it's a dumb watch, I don't care. All cell phones and dumb watches, smart watches, have to be silent or turned off and put in your purse or backpack and leaned up against the wall. When I pass out the blue books, you have to roll up your sleeves so you can show me you don't have a watch on. Are we clear? You want to get the strip, green and white, Scantron strip with 50 seconds on the side. It's all in the syllabus. Get the right one. You can take that to your spot. Write your name on that. Just don't write your name on the blue book. Everybody got it? Then you sit down, your stuff is off, and then I'll have you come down and get your exams. And after everyone is back, and I write a random number on the inside of the cover, I pass out the blue books. Okay? Let's see, I don't know what my exam looks like. Various forms of my exam. Let's see, there's a random one. We'll have something like 27 to 30 2-point multiple choice questions. 20 to 23 matching questions worth 1.5 points each. On average, there will be 3 essays on lecture 1. Could be 2, could be 4. On average, it's 3. Okay, so don't take it to the bank because I have like 8 forms of all these exams. On the preliminary concepts, we had things like specificity and homeostasis, the 2 different types of feedback loops that your body uses for homeostasis. We talked about some principles of the fluid mosaic model of cell membranes. There are 5 different components. There's actually a lot more, but the top 5 I gave you, and you only need the first 2 to show selective permeability. There'll be several questions on transport processes, both passive and active. Gotta know what's moving, solute or water. You do need to know about enzymes. Again, there'll be some visual recognition. You need to visually recognize ATP and how much energy is between phosphate groups. You have to be able to visually recognize fatty acids, triglycerides, amino acids, and small peptides with peptide bonds. You need to be able to visually recognize the difference between condensation and hydrolysis reactions. You also have to be able to visually recognize the 3 stages of aerobic cellular respiration. You gotta be able to visually recognize the difference between purines and pyrimidines, as well as the phosphodiester bond between nucleotide links in a nucleic acid. So peptide bonds between amino acid links, phosphodiester bonds between nucleotide links. You do need to know about the electron carriers, NAD and FAD, which one is the low energy, which one is the high energy, which one is the reduced state, which one is the oxidized state. And you gotta know the difference between substrate level phosphorylation and oxidative phosphorylation and where they occur in the cell. There'll be a couple questions on anaerobic respiration. You do need to know that first reaction of the Krebs cycle. Acetyl-CoA and oxaloacetic acid becoming citric acid. You need to know the difference between lipolysis and beta oxidation and the products of beta oxidation and where they enter into aerobic cellular respiration. You do need to know you do not need to know the chemical structures and formulas of the nitrogenous bases, okay? But you do need to know which ones are purines and which ones are pyrimidines. And just a base, what does a pyrimidine look like versus a purine? Okay? You gotta know which bases are found in RNA and in DNA. You gotta be able to do complementary base pairing, either DNA to DNA or DNA to RNA. Or even RNA to RNA, because that's what happens between codons and anticodons. Read the question with ruthless accuracy. It is straightforward if you have the wherewithal to concentrate on reading the question.
Oh my god.
This is it.
Oh my god, I'm dying.
Sorry.
That scared the shit out of me.
Which, by the way, is an illustration of why you have to silence your cell phones for the lecture. You do not want that going up.
It's on, it was on Firebird, sorry.
All right. Yeah, and you have to do complementary base pairing. Just read it carefully which way you're going. You gotta know the difference between transcription and translation and what type of enzymes and nucleic acids are involved in either. You do need to know initiator and terminator codons. Again, cell division is really good for matching. You got 3 of them. Mitosis with interphase, a cell cycle, and then you got meiosis 1 and you got meiosis 2. It could be any of them. Just know the 3 nutrients really well as well. Okay? Any questions? And then that's all we have next week. Once you turn in the lecture exam, and there'll be 2 piles. There'll be a Monday lab pile and a Wednesday lab pile. Please turn it into the pile that you are registered in. If you have switched sections for convenience and we didn't switch CRNs, it's whatever pile you are registered in, not where you actually do lab, okay? Any other questions on the lecture exam? That's all we got. All right, let's look at the study guide for lecture exam 2. I do want to do a little introduction on this. This is the lecture exam 2 is about the nervous system, basically. It will eventually go to detailed discussions on the functional areas of the brain, peripheral nervous system, and then the senses. Some portions of the anatomy that would be helpful for you for the lecture exam 2 material are the functional areas of the brain that should have been covered in anatomy, as well as the cells of the retina. Oh, and also the inner ear. Okay? Those are 3 areas that will be helpful for you to review from anatomy when we get to those sections. Functional areas of the brain and cells of the retina and the inner ear. So just so that we keep this straight, I'm going to do anything on the whiteboard over there. This is all lecture exam 1 here and here. Okay? All right. Some anatomy review. Remember what a neuron looks like. We have the dendrites, which is an input zone that's getting signals from either sensory receptors or other neurons. We have the cell body. Next to the second bullet, I would write also an input zone. The cell body can also receive inputs from other neurons. Remember that the cell body is where the nucleus is, as well as packets of rough endoplasmic reticulum and ribosomes we call Nissl bodies or chromatophilic substance. Chromatophilic substance and Nissl bodies are gray in color. So next to the second bullet, make sure you write that gray matter is where the cell bodies are. The cell bodies make gray matter gray, because they're gray, bluish gray. The axon is the conduction zone. It may or may not be myelinated. Okay, now, what I would write next to this first slide on lecture exam 2 material is we are going to concentrate on the physiology of multipolar neurons. Like what is written here, multipolar neurons. Unipolar and bipolar neurons do things completely different. So most of what we're going to talk about, especially in the beginning, before we get to the senses, is about this. Multipolar neurons, multiple dendrites, each one is a pole, and one axon. Multipolar, you guys remember multipolar neurons?
Yeah.
That's what we're basically studying here. Here's 2 multipolar neurons. Everybody see how we're going to concentrate on multipolar neurons? Some more anatomy review that I want to get done before Wednesday's lecture. Remember that CNS is the brain and spinal cord. PNS is everything else. This is a gross anatomical distinction. This is not a neurological distinction. Gross anatomical distinction only. The same neuron runs in both. There are some neurons where are only CNS and some neurons that are only PNS, but a lot of neurons run in both. Motor neurons have their cell body in the CNS and the axon in the PNS. It's the same single neuron. Everybody got it? Mm-hmm. And then we're going to look at some of these other terms. So you have some neurons that are sensory, some neurons that are motor. We use some different terms for CNS and PNS. In the PNS, we use afferent for sensory neurons. In the PNS, we use efferent for motor neurons. That'll help you not just for physiology, it also will work for you in English. That's the difference between affect and effect. So, but in the CNS, especially as it relates to the spinal cord, and these are just really easy motor neurons, multipolar. The little eyelashes there, those are the dendrites, the circle is the cell body, and the single line is the axon. And because of that, we use different words in the CNS. In the CNS motor, the signal is descending the spinal cord, so we call these descending. And sensory signals are going in and going up, that's ascending. So get used to some of those terms now. And in here, out in the PNS, they're using the afferent, efferent. And in the brain and spinal cord, it's ascending, descending. We do have to look at the, also the neuroglial cells. In the PNS, the only one you're responsible for is the neural endocyte or Schwann cell. These are the myelinating cells. In the CNS, the myelinating cell is the oligodendrocyte. So this slide is about myelination. Remember, some axons will be myelinated, but not all. Now, one difference between these two is that a neural endocyte will dedicate itself to one axon and myelinate about a millimeter of one axon. Because in the brain and spinal cord there are so many axons condensed, a single oligodendrocyte can branch out and myelinate several axons. This is the faithful spouse. This is the hoe. Now, here's what, I hope they covered this in anatomy. Pretty much, well not all, but pretty much all axons actually do run in these cells, especially out in the PNS. In the PNS, even an unmyelinated axon runs in a Schwann cell neural endocyte, okay? But it's unmyelinated because it just sits in it like this. This would be unmyelinated. When you have a myelinated axon, the Schwann cell is triggered to wrap itself around the axon over and over and over again until it squeezes the cytoplasm into just one part of it. It's almost all phospholipid bilayers. Look at all these lines over here and know that each one is a phospholipid bilayer. That thing is covered with wax. Phospholipid bilayer, when you concentrate it, looks like wax. If it's unmyelinated, it just sits in it, and it's not really covered by all this phospholipid bilayer, just one. Now these are the CNS neuroglial cells. This is anatomy review still, right? We covered this in anatomy, correct? So we already covered the oligodendrocyte, myelinating cell that branches and myelinates several. Then you got the astrocytes, which is actually the most numerous of the CNS neuroglial cells. They're really about the blood-brain barrier. They're underlined. So if you see a neuroglial cell wrapped around a blood vessel, it's an astrocyte. But this is also an important neuroglial cell and injury repair as well. Again, we already covered oligodendrocytes, they branch and myelinate several axons. Because of the blood-brain barrier, neutrophils and macrophages can't get out of the blood vessel to go out to the CNS. So there must be a cell there to do that job, and that's the microglia. They are the fixed phagocytes of the CNS. And again, the reason why is because your white blood cells can't get out, can't get past the blood-brain barrier. If you have white blood cells on your brain and spinal cord, you are knocking on death's door. Everybody got it? And ependymal cells are all about cerebral spinal fluid, CSF. They make it, they monitor it, and they circulate it with these little cilia on the top. So they look like that. Okay, everybody got the neuroglial cells? This is a review still of anatomy. In fact, the picture looks like the one in anatomy, don't it? All right. Here's now we're going to start getting into some physiology. We already covered for lecture exam 1, it's one of your essays, the different ions in the ICF and the ECF. Does this ring a bell? And remember, ions cannot cross the lipid bilayer, they have to go through protein channels. So here's a review of them. Outside we have more table salt, sodium chloride, and calcium. Inside we have more potassium. We also have some other ones like phosphate and other things. But for lecture exam 2, you also have to know that proteins, when they are in solution, tend to be negatively charged. So positively charged ions are called cations, and negatively charged ions are called anions, and proteins are one of them. There are many other ones, but that's what it's saying here. If you've got proteins in the cell, it's going to be more negatively charged. Now this term here, I want you to get past the strangeness of it. When we talk about membrane potentials, you want to translate that to the voltage difference across that membrane. And I had mentioned this initially when I talked about the sodium-potassium pumps that you got to know for lecture exam 1. An example of primary active transport. You guys remember that? Sodium-potassium pumps? And I said 3 doesn't equal 2. 3 sodiums out, only 2 potassiums in. That is one of the reasons why your cells are more negative on the inside. You got 3 outside, 2 inside, it's more positive on the outside. So when we talk about membrane potentials, it could be said in 2 ways. It is more positive on the outside, or it's more negative on the inside. It's still a voltage difference across that membrane. Everybody got it? But even though we could say it 2 ways, we always talk about it what it is on the inside of the cell. So we will talk about it in negative numbers. Everybody got it? All your cells have a negative membrane potential on their plasma membrane. All cells have a negative membrane potential across their plasma membrane because 3 doesn't equal 2. Okay? And I think we're gonna stop there.
Because
Put this up in case you wanna fill in those blanks. This is where I'll start on Wednesday.
It's because of the sodium potassium pumps, because it takes 2 in and 3 out, so 2 is